All right, so the title of our paper was Tau interactome maps synaptic and mitochondrial processes associated with neurodegeneration. And so I'll begin by giving a little background on Tau, which is the focus of this work. And so Tau was identified kind of a long time ago, and when it was first looked at in neurons, it was found to be a protein that binds very strongly to microtubules, and it actually stabilizes microtubules. So it was called a microtubule associated protein back then, and it still is. However, since then, there have been a number of studies showing that Tau can bind to other proteins in the cell, including actin and other cytoskeletal proteins. So it's a bit more complicated than just a protein that binds to and stabilizes microtubules, is what we're realizing in more recent years. And the reason we're interested in Tau is because there's a class of neurodegenerative diseases called Tauopathies, which are characterized by the accumulation of pathological Tau in the brain. And here's just a list of the diseases, with obviously Alzheimer's disease. Maybe, I don't know if you can see my pointer, maybe Alzheimer's disease being the most common Tauopathy. And then we have, below that, we have other Tauopathies that are also characterized by Tau accumulation in the brain. And we think that in these Tauopathies, that Tau is the important protein that is driving the disease, because one set of these diseases called familial frontotemporal dementia is caused by mutation in the gene that encodes Tau. And this is just showing part of the MAPT gene, where you can see the different mutations in the gene that have been identified that cause FTD in humans. And so this is, important evidence that just a single point mutation in the Tau gene can wreak havoc in the brain and cause dementia. And so that leads to this model of this gain of toxic function effect of Tau in neurodegenerative Tauopathy, where especially in later age, there is an accumulation of this toxic Tau in the brain, and this could lead to pathophysiology and neurodegeneration, which underlies the cognitive impairments that people suffer from. And so this is a model that a lot of the field is following. And but the major questions that we still have is, how is Tau causing pathophysiology, and how is Tau causing neurodegeneration? And these are really important questions because if we can figure out those mechanisms, then maybe that could be targeted for therapeutic purposes. To recover some cognition in the disease. And so how do we know that Tau is affecting neuron function at this point? Well, there's been a lot of literature on this showing that Tau can actually have an effect on various aspects of neuron function and through multiple mechanisms and even protein interactions. And here's a few important ones. Tau can affect cytoskeletal dynamics in cells and axonal transport. Also, the function of the axon initial segment, which is important for action potential generation in the neurons. And then there's evidence that Tau affects nuclear transport and proteostasis. And then, of course, Tau can affect synapse function. There's a lot of literature there. And so how is Tau doing all of this with this gain of toxic function effect? This is a question that I've really been fascinated by for a number of years, and a lot of people in the field. And that's kind of what we're trying to get at with this paper. How is Tau having these effects in the cell? Another interesting thing about tau and disease is that there's kind of this stereotypical spread of tau throughout the brain in humans as it accumulates. Here is an example of Alzheimer's disease. And so the blue labeled here is where the pathological tau is in the brain. And you can see with the progression of time in disease, this blue signal is spreading throughout the brain. And there's evidence in the literature that the tau is actually spreading across neurons that are synaptically connected, kind of in circuits through the circuitry. And so there's been some studies showing that tau can get released from a presynaptic neuron and get taken up by a postsynaptic neuron. And it's possible, we think, that the activity-induced release of tau across the neuron circuits can promote the spreading of pathological tau in the brain. And so there's some literature supporting this. However, it's not clear yet how tau actually gets out of the cell to start the spreading process. So this is another interesting question that we found some clues to in this paper that I'll tell you about. With that introduction, I'll get into the paper. And I want to point out here that this was a huge collaboration with many people making very important contributions. So I want to give a shout out to my co-first authors, Jesus Madero Perez, who did the work on mitochondrial bioenergetics that I'm going to show. And then also Danielle Sweeney, who is an expert in mass spectrometry, and she's at UCSF currently. So she, made these significant contributions to get this mass spec data for this paper. And then also, of course, a shout out to Li Gan, who was my mentor in my postdoc and who supported this work and provided the guidance he needed to complete it. So, and of course, shout out to everybody else who made important contributions. For the paper, I just want to break it down a little bit, and I'm going to talk about two parts for this paper. The first part is our findings that suggest a potential way that tau could get secreted from neurons during activity. And the second part is more looking at that question of how is tau causing toxicity in neurons. And this is our other kind of schematic showing the two different parts that I'll go over. So the approach in part one is to take neurons that have wild type tau, and we stimulated those neurons to induce their activity. And then we used a system called the APEX, which is a proximity dependent ligase, to identify the proteins that were interacting with tau during the increase in the activity. And then the part two is where we compare wild type tau to tau with mutations that cause FTD, two of which, P3ONL and V337M, you may have noticed in my previous slide of all the mutations that cause FTD. And in this approach, we used the affinity-Purification mass spectrometry approach or method that allows us to identify a large number of tau interacting proteins. And we think this will give us clues about the toxicity mechanisms in disease, in neurons in disease. So I'll start with part one, and this is the APEX proximity dependent labeling of tau interacting proteins. And how this works is shown in the schematic. So we start out with a human tau that is fused to the APEX2 enzyme. And when we add biotin to the neurons, it gets taken up into the neurons. And then we give a very brief application of hydrogen peroxide for 1 minute, and that stimulates the activity of the apex enzyme, which will start to biotinylate the proteins that are in close proximity. And when I say close proximity, it's estimated that this is within 10 to 20 nanometers of the apex enzyme is where other proteins will get biotinylated. And these proteins get biotinylated on the tyrosine residues. So you see with the little red dots will represent the biotinylation of these proteins that are close to the apex on tau. This labeling is done in live neurons. And after the labeling of the tau interactors is completed, then we lyse the cells. That's shown here. So we lyse the cells. We have non-interacting proteins that are not biotinylated, and then we have proteins that are tau interactors that are biotinylated. Then we trypsinize the proteins and use antibiotin pull down to extract the biotinylated proteins, and followed by an elution. And we do the proteomic analysis on those. So this is a very nice technique that was developed by Alice Ting's group in this paper, Idushi et al. From 2017. So the advantage of using this approach is that we can do the labeling of the interacting proteins in living neurons that have intact cellular compartments. And I think we all know that neurons are very compartmentalized. And so this is important to look at the interactions in the kind of endogenous compartments of the cell. The other cool thing about it is that it can provide a snapshot of the tau interactome in time. So we only do a 1 minute stimulation of the apex enzyme. So whatever gets biotinylated is only interacting with tau in that 1 minute of time. It also allows for the labeling of both transient and more stable interaction partners, whereas more conventional approaches tend to pull down just the stable interactors. It's potentially that there's a potential that we identified some proteins that are maybe only interacting with tau very quickly. So in terms of how we set up the experiment in neurons, we expressed 2 versions of apex tag tau in neurons that were derived from human-induced pluripotent stem cells, or iPSCs. And the 2 versions are, they're both the 2N4R version of tau, which is the longest isoform of human tau. And one version is tagged with apex and a flag tag on the N terminus, and the other version has apex on the C terminus. And the reason we did this was because, at least in part, because the tau protein is estimated to be about 60 to 70 nanometers in length. And, I mentioned that the apex can biotinylate things within 10 to 20 nanometers. So it's possible that we could get some spatial resolution in terms of the interactions in the N terminus versus the C terminus of the protein. Also, tau can be cleaved and at different points in the protein. And so it's possible that, for example, if the C terminus gets cleaved off, that having the apex tag there could identify different interactions of the cleaved product. So we figured it would be cool to look at the interactors on both sides of the protein to see if we could find differences. And then we also made an apex tubulin cell line, and this was as kind of a control for proteins so that we could look for a tau specific interactors. And I'll show you how that works in the data on the next couple slides. And so we took these 3 different cell lines that have these transgenes to express these different constructs and use a neurogenin 2 transcription factor to drive their differentiation into a population of very homogeneous glutamatergic neurons. And this is a method that Lee's group had established a couple years earlier. And it's a very robust method of making neurons. And so here we have a picture of the neurons expressing the apex tau. And this is just showing that we get apex tau expressed, let me see, expressed up here in the cells. And the red signal below it is a staining of the biotinylated proteins, the proteins that are biotinylated by apex tau in the presence of biotin phenol and hydrogen peroxide. When you do not add a hydrogen peroxide to stimulate the apex protein, then you see no biotinylation signal. So we're really switching on the biotinylation of the proteins in that 1 minute of time with the hydrogen peroxide application. We also confirmed the activity of the apex in the cells using Western blot on the right. And so we compare the apex tubulin cells to the 2 versions of the apex tau construct. And I just want to point out something important here that in the flag blot, so this is a blot of the flag tag on the apex constructs. You'll notice in the tau groups, there are actually multiple bands. And the pattern of the bands differs between the N apex tau and the C apex tau groups. And we think this is because there's differences in the cleavage products in these cells. So it's interesting because, for example, in this N apex tau group, this is a 37 kD, cleaved version of tau with just the N terminus and the apex fused to the apex protein. So, the fact that we get cleavage, in the next group, number of slides that I'll show you, these cleaved products could be contributing to the interacting proteins that we identify. And then we also confirmed here that with the addition of biotin phenol and hydrogen peroxide, that's required to get the biotinylation here on the streptavidin blot. You need both of those in order to get that activity. So move to, there we go. So we set up this experiment first to look at the interactors of the N and the N and the C tagged apex tau compared to tubulin to confirm our specificity. And here we have a Venn diagram showing the overlap between these 3, the interactors identified from these 3 groups. And the first thing we noticed is that the apex tubulin group down here strongly overlaps with the tau interacting groups. And so this was very nice to see because actually the GO analysis from these 14 proteins that overlapped among tau and tubulin was primarily cytoskeletal proteins and proteins in the microtubule cytoskeleton specifically. So this is confirming that we're seeing very strong, we have very strong spatial resolution with this technique. That tau and tubulin are both highly associated with the microtubules. So that was very nice to see. And then the next thing we looked at was how does the N-apex tau interactome and the C-apex tau interactome overlap? So 55% of the total tau interactome here that we identified was in both the N and the C tagged apex. So there was quite a bit of overlap, but that also means that 45% did not overlap. So I'll show that next. But here, these 136 proteins we identified, let me go here. The Go analysis on that brought up, again, a lot of cytoskeletal proteins and also known tau proteins, known tau binding proteins, including alpha-synuclein and HSP90, HSP70, and a couple others. So that was also nice to see that we confirmed interactors that have already been shown. And then when we look at the proteins that are different between the N-apex and the C-apex tau, we found some interesting things. A bunch of them in the N-apex tau group were involved in the docking of vesicles at the presynaptic terminal. Whereas the C-apex tau was more involved in or proteins that are involved in the vesicle fusion. So they're both localized at presynaptic terminals, but are involved in different functions. So that was interesting. And then there were a number of other groups that were classified that were different that I think could be explored for future studies. And the next thing we did using this anti-biotin pull-down approach was to actually look at the tyrosines, the specific tyrosines on the tau interacting proteins that were biotinylated by tau. And this is a really cool trick using this approach, I think. So this slide is just showing these tau interacting proteins and how the apex tau is biotinylating them on a specific site. And so using our technique of the enrichment for biotinylated peptides, it kind of actually enables us to identify which residues those are on the proteins. I think we identified over 600 biotinylated residues across all the proteins that we found. And the overlap is here. Again, almost half, so 45% of the biotinylated tyrosines were common between the N and the C apex tau. But you can also see there's quite a few that were different between the two versions of tau. On the right, I have mapped out the tyrosines on these proteins that were labeled by the different apex constructs. So you can see, even for example, in tubulin, that the tyrosines that were labeled were common across all three cell lines. So that's kind of interesting. That suggests that, tau and tubulin are interacting with the same kind of places in the protein and labeling the same tyrosines. And that's also especially evident for MAP1B, where you see this cluster of biotinylation within that protein from both tau and tubulin constructs. So this is kind of nice that we were able to get this very fine resolution on the binding area. We also looked at those presynaptic proteins that we identified that were biotinylated by apex tau. And I have that mapped out here. On the left is an example of a synaptic vesicle that is docked at the presynaptic active zone. And you see all these proteins that are associated with the vesicle, and many of which are important for the fusion of the vesicle during synaptic vesicle release, or sorry, neurotransmitter release. And so the little stars represent, on each protein, represent where the biotinylation from apex tau occurred on these individual proteins. So you can see many of them, many of these proteins have been biotinylated. So, I would interpret that to mean that, tau is, in this sub-region of the cell and very present at where vesicles are being docked at the membrane. And on the right here, we have just maps of the individual synaptic vesicle associated proteins. So SNAP25 and syntaxin-1 are two of the three SNARE proteins that actually mediate the fusion of the vesicles. So both of those were biotinylated. And then we also have MUNC18, which binds to syntaxin-1, and it's required for the vesicle fusion at presynaptic terminals as well. And MUNC18 actually had 6 biotinylated residues, which was actually quite a lot. So that was interesting. MINT1 was also biotinylated, and it binds to MUNC18. So there's kind of like this complex, of SNARE associated proteins that tau is biotinylating. So that was quite interesting. The other interesting thing here is that when we looked at the vesicular, the integral proteins in the vesicular membrane, we did identify a few biotinylated tyrosines on 3 of the proteins. VGLUT2, SV2A, and synaptogyrin-1. And all of these biotinylated tyrosines were on the cytosolic side of those proteins, rather than on the luminal side or inside the vesicle. And we thought this was interesting because it kind of suggests, together with the SNARE complex results, that tau is more associated with the outside of the vesicle rather than being in the inside of the vesicle. And then we also confirmed using staining and a dye called FM143, we confirmed that tau is present at presynaptic terminals in our neurons, in our human neurons, at sites where vesicles are being released. And so what we did here is use this FM143 dye, which when you add it to neurons, it's a fluorescent dye that gets taken up into vesicles that have been endocytosed during activity. And so these green puncta here that I have the arrows pointing to represent the sites where synaptic vesicle recycling is happening at presynaptic terminals. And we see that tau is colocalized with that in many places for these neurons. So it seems to be in the right location for these interactions to occur. And so having found this result, we then wanted to see if we induce activity in the neurons and cause tau to be released, what is changing with the interactions? And could this give us some clue about how tau is spreading across synaptic connections? And so in our human neuron cultures, the first thing we wanted to confirm is whether we could see the activity-induced tau release. And this has been shown in other culture systems, but we used an ELISA to measure tau that has been secreted into the media from the neurons. And we stimulate, depolarize the neurons with high potassium chloride in the external solution. And you can see this in this graph on the left. There's a significant increase in the percent of secreted tau with the addition of the high potassium chloride to depolarize the neurons. This is blocked by BAPTA-AM, which is a calcium chelator that gets into the cell and chelates the calcium that's required for vesicle release. So in this condition on the right, there's no the vesicle release at the presynaptic terminals would be blocked. So this shows that the tau secretion is also blocked when vesicle release is blocked. And this has been shown by others in the literature. We did a control to look at LDH release from our neurons to make sure that the potassium chloride treatment was not killing our neurons, and that was not the cause of the increase in tau in the media. And so you can see across the groups with the potassium chloride treatment, there was no difference in the LDH release. So we do think that this is driven, by the activity in the neurons rather than cell death. So we then applied this potassium chloride depolarization to our neurons expressing the apex tau constructs. So we have 4 groups here. We have the unstimulated neurons with either N apex or C apex tau. And then we have the same groups, but with or following 30 minutes of high potassium chloride treatment to induce neuronal activity. And then we stimulate the apex with hydrogen peroxide and pull down the biotinylated proteins in each group and do mass spectrometry on those. And you can see here on the right of the Venn diagrams where the blue represents the interactome identified in the unstimulated neurons, and the red is the interactome identified in the neurons that were depolarized to induce activity. And the first thing you notice is that it's mostly overlapped. So most of the interactions in the neurons do not change with activity, the vast majority of them. However, there are a few that do, and most of them were in the were increased interactions in the neurons that were depolarized. So there were fewer. You could see there were only 15 and 9 that were only in the unstimulated groups. So we focused more on the hits that were increased interaction with the depolarization. So these 44, there were 44 in the N apex tau neurons and 69 in the C apex tau neurons. And then we also looked at the biotinylation sites on those as similar as I showed you before. So here we have the analyses of the interactors that were increased with the depolarization and increased neural activity. And so one of the main groups that came out in from both interactomes were proteins involved in synaptic vesicle release and glutamate secretion for both sides. So this we thought was very interesting as it relates to the release, the release of tau from the neurons. So we followed up a little bit on this, but there are also other interesting things here from these analyses that we have not yet followed up on. So I think there's, potentially more here that could be looked at in terms of how tau is changing with neuronal activity. But in following up on this, the changes at the synaptic vesicle associated proteins, here we have the maps of the where some of these are biotinylated, and a really interesting one that came out was synaptotagmin 1 on top here. So there were 2 activity-dependent biotinylation sites on synaptotagmin 1, and this is the calcium sensor. It's a synaptic vesicle protein, so it's in the synaptic vesicle membrane. When calcium binds to the C2a and C2b domain of synaptotagmin, this is when synaptic vesicle fusion is triggered from the cell. So this was an interesting finding, that this was actually activity dependent, the interaction with tau. And then again, we saw some additional sites on MINT1, which is associated with the SNARE complex that were activity dependent. And then some other synaptic vesicle membrane proteins, SV2C and synapsin 1. And we confirmed, we used an approach called proximity ligation assay to confirm the interaction of tau with synaptotagmin 1. And this assay makes use of antibodies. So we took an antibody that recognized tau and an antibody that recognized synaptotagmin 1 and co-immunolabel our neurons, our human neurons. But then you use a kit that allows you to fluorescently label these 2 antibodies that are in very close proximity. So I think they have to be less than 40 nanometers. The 2 antibodies need to be less than 40 nanometers apart in order for you to see signal from the proximity ligation assay. And that signal is this red signal in these images. So here you can see we have in unstimulated neurons. We did see some PLA signal between the tau and synaptotagmin 1, suggesting there is some interaction there at the unstimulated that we can detect using this approach. However, when we depolarize the neurons with the 50 millimolar KCl for 30 minutes, and then do the proximity ligation assay, we see a much increased signal, suggesting that there is the increased interaction of tau synaptotagmin during the enhanced activity. That's quantified here. And we have a control where you do the proximity ligation assay with only one of the antibodies and not 2, not both, showing that you don't get any signal if you don't have both antibodies there. So the summary of part 1 is that we found that tau is associated with cytosolic side of synaptic vesicles and with the SNARE complex. So it kind of positions tau to be at the sites where it could be released. We also found that neural activity increases binding of tau to synaptotagmin 1 and other vesicular proteins. And we think this could be pointing towards a mechanism for how tau gets released from neurons during the activity. And the really interesting thing I still wonder is how is tau getting out? It's not inside the vesicles, or it doesn't appear to be inside the vesicle. So, when the vesicle docks and the SNARE proteins pull apart the membrane at the presynaptic terminal, the membrane is compromised. It breaks during the vesicle fusion event. So I've always wondered, is it possible for tau to penetrate the membrane during that fusion event when the membrane is compromised? So, we haven't seen any evidence for this yet, or this hasn't been shown, but I think it's an interesting idea for potentially how tau could be getting out with this mechanism. So now I'll get into the second part for the last part of the talk. And this is to look at the interactome of the wild type tau versus the FTD mutant tau. And for this approach, we used the affinity-Purification mass spectrometry to identify as many interacting proteins as possible. So we were able to pull down more with this approach than with the APEX approach. Excuse me. So here we had, again, 3 different cell lines. We had the wild type expressing, the tau wild type expressing cells, and then cells with V337M mutation and cells with P301L mutation. We expressed these in neurons, and then we performed a lysis of the cells. And following the lysis, we pulled down the flag tagged tau constructs by IP and eluded the tau interacting proteins and performed mass spectrometry. Now here we have a Venn diagram showing the changes in the groups. So we're comparing the mutants here, the FTD mutants, to wild type tau in this diagram. And surprisingly, we found that most of the differences were showing that wild type tau was interacting with more of the proteins than the mutant version. So there was almost kind of a loss of interaction with the mutant tau. And so we ended up focusing mostly on this data where that says up here, which were the ones that were more strongly bound to wild type tau than the tau mutants. We did identify some that were downregulated, less in the wild type compared to the mutant. So the mutant would be binding to these proteins more. But we didn't focus on that in this paper. I do think there are things that could be followed up on in future work though. So, that's part of the reason this was a resource paper, is because I think there's a lot more in this data than what we completely dug into here. But focusing on these ones, the most of the interactions that were stronger in the wild type versus the mutant, the Go analysis on these hits were showing very strongly that actually wild type tau was more strongly interacting with mitochondrial proteins, shown here, and but also with ribosomal proteins. And so we, for the rest of the paper, we really focused on these hits as kind of a strong effect that our data showed. So in terms of the mitochondrial proteins, I think most of the mitochondrial proteins that were pulled down with tau were in the inner mitochondrial membrane, shown here, and the electron transport train. So you can see the different colors we have depicting here are the hits that were greater, where's my mouse, that were greater in wild type tau compared to either V337M in green or compared to P301L in purple. And then we also identified a few that were interacting with wild type more than both of the mutants in blue. So most of them were in the inner mitochondrial membrane, but we also saw some interactors in the intermembrane space and in the outer mitochondrial membrane. So throughout the mitochondria, we identified a number of tau interacting proteins. So we then wanted to validate some of these interactions using another approach. And here, we actually used a different set of cell lines, a different set of IPS cells. And I'm showing them on the top left here. We had cells that were from a control individual, and they're expressing endogenous wild type tau. And then we used CRISPR edited versions of those cells that had a FTD, the V337M mutation introduced into the same genetic background. So they're isogenic. And we had both heterozygous V337M mutation and a homozygous V337M mutation. So across these three cell lines, we used a proximity ligation assay that I described earlier to look at, with a tau antibody and the antibody to a mitochondrial protein to look at the signal, the PLA signal representing where these two antibodies are in close proximity, suggesting the interaction between tau and the mitochondrial protein. So we looked at three different ones, and the staining is shown on the right here. But I'll point to the bottom where the quantification is. So ATP 5IF1 we looked at this inner mitochondrial membrane protein. And you can see in both the heterozygous and homozygous V337M neurons, there was a decrease in the interaction measured by PLA. We showed a similar effect on TOM40, which is in the outer mitochondrial membrane, and also with TIM13, which is in the intermembrane space in mitochondria. So this effectively validates what we found in the mass spec results. We next went on to see whether the mitochondria are affected in these tau V337M neurons, which could be associated with the changes in the interactions that we see. So we did a number of experiments to assess mitochondrial function in these cells, and I'm just showing some of it here. This is fluorescence from TMRM, which is a method to look at the mitochondrial membrane potential in the neurons. And again, we looked at the three genotypes here that I mentioned on the last slide. And you can see there's a significant change in the TMRM intensity in the cells with the V337M mutation, suggesting there is an effect on mitochondrial function in these cells. However, the number of mitochondria is not different across the cell types. And this is some more analysis looking at the bioenergetics of the mitochondria, excuse me, in the neuron cell lines. And we did experiments when the cells were both 2 weeks old and 4 weeks old. So you can see there's reduced levels of coupling efficiency, respiratory control ratio, and spare respiratory capacity in the neurons that have the V337M mutation. So all of this suggests that the energetics in the neurons with the tau mutation are affected, and the mitochondria are not as able to produce sustained energy as the wild type neurons. And lastly, we then looked at these tau mitochondrial interactors in a human cohort. So this data is from a larger study that showed that there are 6 co-expressed protein modules that are associated with Alzheimer's disease. And this study characterized those modules, Swarup et al. From 2020. The data comes from 3 cohorts of proteomics data. I'm just going to show the banner results here. But what we found was that across these AD-associated protein modules that were identified, we found 29 of our tau wild type interactors. And when we looked at the protein levels across the human subjects, the AD subjects had reduced levels of these tau wild type interactors. And the levels also correlated with the severity of the neuropathology, both by SARAD score and BRAC scoring. And so this is for the wild type versus V337M interactors, and then the bottom is compared to the P301L mutant. this human data set is also great because, I mentioned 6 modules. Well, we looked at one specific module within this data set that included a lot of the mitochondrial electron transport proteins. And that's shown here on the left. This is the module from the paper showing the association with AD. And in the colored circles represent the hits from our study, the tau interactome, where these hits were interacting more with wild-type tau than mutant tau. And when we look at the data in the human subjects, these hits were across the board reduced in the human brain and associated with pathology. So we think these interactors could be, physiologically relevant in human Alzheimer's disease and other tauopathies. So in summary, I talked about 2 parts of this paper, mainly the findings that we had related to activity-induced tau secretion, and that tau could be associated with the SNARE complex proteins and synaptic vesicle associated proteins and get out of the cell with the induction of activity that way. And then the second part is related to toxicity mechanisms triggered by tau. And we found this weakened interaction of tau with mitochondrial proteins that we think is associated with impaired bioenergetics. And I just want to acknowledge, of course, the people in my lab. I started my lab at the Buck a couple years ago. And I did work on this partially while I was in my new lab, and Grant Conway from my lab helped with that. But I also, again, want to acknowledge the support of Lee Gann, who was my mentor, and her, her guidance was so important for this work. Jesus Madera Perez was in her lab. He was my co-first author on this, as well as Danielle Swaney, who is currently still at UCSF with her own lab. She did all the mass spec. And then to acknowledge Dan and Tim at UCLA, who helped us with the human proteomic data and looking at our data set in their work. And Virginia and John and our funding sources that supported this work. And I'll finish there.
Thank you so much, Tara. No, it's a wonderful presentation. Learned a lot. If anyone has any question, feel free to ask through chat, or if you'd like to raise your hand and unmute yourself. I can start with the first one, Tara. I think the result on mitochondria is super interesting, but I think your results on all the translation, initiation, and ribosome also really caught my eye. So I'm wondering, for those with increased tau binding during neuronal activation, do you think they're just increased in translation initiation overall, or is there increase actually more specifically to binding to tau for those kind of proteins? And what do you think about the overlap that you see kind of between the ribosome and the translation initiation in those 2 different experiments?
Yeah, so that's interesting. Yeah, we did get a lot of RNA binding proteins across the board in both interactomes. And we did do some analysis of comparing both interactomes that we included in the paper, and I think would be important to follow up on, but we haven't done that. In terms of the translation initiation factors, yeah, I think this is an interesting question that we also haven't looked at for this paper. My lab is currently looking at one of the hits that we pulled down in this study, and we think that tau can actually inhibit activity-dependent translation. And we don't have the mechanism quite fleshed out yet. But there's also other, in the literature, there is other reports that tau can affect translation overall, right? Through either stress granules, or maybe even other mechanisms, depending on the circumstance. but yeah, I think it definitely, it's an interesting question, and I'm glad that we saw a bunch of hits in our study, you know, because it kind of goes along with what other people are finding of tau interacting with RNA binding proteins.
Right, yeah. And in the chat, we have a question from Anastasia asking, why do you think there's reduced tau interaction in AD?
Reduced tau interaction. Yeah, so we think that wild type tau, or sorry, tau in, you know, normal tau is interacting more with mitochondrial proteins than maybe tau in disease, or at least with FTD mutation. That would be specific to the mitochondrial proteins. We did identify a few proteins that interacted more with tau when tau was pathogenic. So we did identify some of those. We just didn't follow up on it in this paper. So it's certainly something in future, you know, that there could be some interesting proteins that interact more with tau and disease. It's just for our work, we identified more of them in mitochondrial that were associated more with the normal tau. I hope that makes sense.
Right, and just like a little follow-up on that, I guess since you use iPSC, the strength is that you might be able to see clinical correlate. So I wonder if you see any difference in disease status, stage, gender.
Oh, for interactions?
Yeah.
Oh yeah, we haven't looked at that. That's a good point. And yeah, we did not get to that level with this paper. But I think the cool thing is, so even though we're somewhat limited in terms of the iPS cells for answering that, you know, that's where the strength of the larger human proteomic data set comes in. Because, you know, I can't remember off the top of my head how many subjects were involved in that Banner study. But, you know, the fact that our tau interactome proteins, you know, showed effects in this larger Banner cohort of people, you know, makes it, I think, likely that they're important, you know, the proteins that we identified in our single kind of cell study.
Right. Okay, I think we're running a little short on time, but I think if anyone have any question, I'm sure we have posted Tara's info. So if anyone wants to reach out to Dr. Tracy, you can through her websites and our information. Our partners at ADDI also have this AD Connect platform for people to interact there. And thanks everyone for attending and listening. And thanks so much, Tara, for the great presentation.
Great, thank you, Kuan. Thanks very much.